Kira Koto. As to paraphrase the Lorax, I'm Maj, and I speak for the fungi. So I'll just get started. This is work that my master's student Marley Ford put together with an honor student Manisha Prasad, and then the rest of the crew of the old fogies doing some of the other not very exciting work in the lab rather than in the field. So Rama, Rowan mentioned this earlier. It's Lophomyrtus bullata. It's a New Zealand endemic, and it often forms a shrub to a small tree. It ranges from the top of the North Island of New Zealand to the northeastern corner of the South Island. And it was once a common tree, but it has recently, as Ron mentioned earlier, has been given the threat status by the Department of Conservation as as critical and nationally threatened. And of course, surprise, the reason is because of myrtle rust. And this was one of the first trees that was impacted in the wild when Austrobruxania sedii came to New Zealand. And it has, really, really impacted this plant. And as you can see, the distribution of Lophomyrtus bullata is basically the distribution of where myrtle rust is. So where you find the plant, that's where you're going to find the rust as well. And so since it's a threatened tree, one of the questions we had is that, well, now what? Of course, it's like one of those things is that nobody actually recognizes it or thinks about it until it's threatened. And one of the things that we had no idea about is what's the associated biodiversity associated with this tree. We don't understand its ecology, and certainly we know nothing about its fungi. So Marley, he did his master's, finished about a year ago, and he was particularly interested in looking at what fungal isolates, what fungi associate with this tree. So you can see it. Oops, sorry. Go back. Over here, this is it. So usually, when people look at it, it's this bubble leaf, and it's that you can see over here. It's with myrtle rust on it, but it can grow fairly tall, which is pretty interesting. So he looked at 3 different locations, all in the North Island, and this was sort of during in between lockdowns. This was sort of his home base, and so he had 3 different locations in 3 different forest types at the northern tip of North Island. And he took soil samples, and he also sampled 5 trees per locations for a total of 15 trees. And each of these sites, he also recorded the associated vegetation and ecology and just general characteristics of the soil. This was one of the locations that he did his collecting at, and he was very happy to send me this picture while I was stuck in Auckland in one of the lockdowns, saying that, I wish that you could come out with me too. And I said, yes, I wish I could have come out with you too, except I'm now stuck in Auckland, not being able to leave. Anyway, but he had a good time collecting. So he collected, as I mentioned, 5 different root samples from 5 mature Rama trees, and then he used amplicon sequencing to characterize the fungal community. And he wanted to look at both the arbuscular mycorrhizae and also the different other fungi that are associated with it. And then together with his work, Manisha was she was an honor student, and she was looking at fungarium records. So she examined all of the different fungarium records of fungi, and also looked at herbarium records to look at herbarium sheets to see whether she could find any associated fungal diversity. Also, there were 8 targeted surveys just to see what other organisms are associated with this tree. So this is just a root section in which you can sort of see some fungal staining, and I have another picture of it next. So one of the things that he found was that Lophomyrtus bullata does associate with arbusculum mycorrhizae. This might not be too surprising because there was a study in which they had hypothesized that this entire tribe of Myrtaceae did associate with arbusculum mycorrhizae, but he was the first person who actually was able to look at the roots and be able to look at these structures. So you can see that this is these are some root cells, and you have the arbuscule in there, and then a coil as well. What was really interesting about it was that there were 7 arbusculum mycorrhizal families that were associated with this one mid-storey tree. And it was really surprising because there was a huge diversity of Archaeosporiaceae. Archaeosporiaceae is a family within arbusculum mycorrhizae and Glomeromycota, and it's not usually found in very many places. And in fact, when he looked at the diversity of sequences that he isolated from these, they were 15 trees. It represented the entire diversity of this family, which was just incredibly surprising. The other thing that was really surprising was that there really wasn't very much in terms of Glomeromycota, which is another family within Glomeromycota. The reason why this is also surprising was because the northern tip of the North Island, that's usually where you have kauri forest and Agathis australis, and they associate primarily with Glomeromycota, and it was only with one species of Archaeosporiaceae. So just like in the same sort of location, 2 completely different families of arbusculum mycorrhizae. And then the other thing that he found was that the species composition of the vegetation was very similar, but the diversity of the fungi changed, but the abundances changed, but the species composition was the same. Even though the trees and the plants were the same, you still had this diversity of fungi. So the other thing that he looked at was ITS sequences. So this was just to look at the other fungi, because we know that, of course, within roots you can have mycorrhizal fungi, but then you also have all of these other endophytes that we just have no idea what they are and what they do. This was one that was very interesting. This was the Archaeorhizomycetes. And this is something that was only discovered about 12 years ago or so. This group was described, and it had originally been only found in a sequence, and then they were able to culture it, and which was why they were able to name it. And so it was really interesting to see that one of the most abundant groups within the ITS sequences was this particular group. And again, very similar to the arbusculum mycorrhizal graph that we saw. You have, in the different locations, you have the similar species compositions, but different abundances of these species. So as I mentioned, together with what Marley was doing, Manisha was looking at field surveys and herbarium records and fungarium records. And so from the surveys of 3 different locations, they found 95 lichens, which is, again, this incredible diversity of fungi that are associated with this tree that we had no idea about before. Looking in the fungarium, there were only 18 collections. Again, this is something that really wasn't looked at before. But it did have this basal Poxenia mycotina, which is Sacosoma album. It's just like this little dried crust-like fungus on wood. So it's really interesting to see that you have this, wealth of fungal diversity associated with this one tree. The other thing, this was during a disease assessment that James is going to be talking about tomorrow. That was an Ophiocordyceps, and we were turning these leaves over to see what the pustules were, found this little Ophiocordyceps just on the back of it. So, looking at the soil characteristics, the 18S, which was the arbuscular mycorrhizae, the variance the pH explained only 12% of the variance, and nitrogen about 13%. So it's still really puzzling, like what actually is driving the difference between these different communities, and is it the plant that's actually influencing the community composition or something else? I don't know. So just to conclude, Lophomyrtus bullata associates with a multiple species of fungi and a really high number of arbuscular mycorrhizal fungi and lichenize fungi. And we do need to do further work to see whether any of these fungi actually solely depend upon Lophomyrtus bullata, or whether they're generalist and we would find them in other places. Because of course he was only looking at this one species and not at all of the other species. So why is this different? Is one, we want to record what fungi are present before they all lost, because we lose all of the trees. And then, there's genetic resistance, but there could also be some resistance conferred by the mycoflora. So if we can identify what the mycoflora are that are associated with these threatened plants, maybe we can use that in breeding resistance. Put the good fungi back in there to be able to save the plants. So just to acknowledge Matthew Calder and Brian from Burton Conservation, who contributed data, and then landowners in Tararua to allow access to these areas. So thank you.